I work about the intrasolar delivery of antibodies to target intrasolar targets using nanomedicine. This is a very important and challenging thematic since, as you may know, there are hundreds of different antibodies being developed in clinical trials and also already in the market that were designed to interact with a wide range of extrasolar receptors. But here the question is, what about the intrasolar targets? In fact, the intrasolar targets represents around 50% of the human proteome. So here we have a huge window of opportunity, because if we achieve to deliver the antibodies inside the cell in its active form, there are thousands of potential therapeutic targets to be explored. What is an excellent strategy and a new opportunity to the undruggable targets, for example. However, the intracellular delivery of antibodies is tricky because by their own they are incapable to cross the cellular membranes, and it's also challenging to develop systems that efficiently deliver the antibodies intracellularly and that are stable in vivo. So in the last years, different techniques have been proposed for the intracellular delivery of antibodies. In this figure, you have a summary of the most used ones. Some of the most common ones are the use of viral vectors or cell internalization peptides, but all of these techniques have some advantages and disadvantages. In the case of virus, the immunogenic issues, and in the case of cell-penetrating peptides, the fact that the antibody is chemically modified, which can affect its functionality. So in our case, we are going to use nanoparticles to encapsulate the antibodies and promote the intracellular delivery of them. And we are using polymeric micelles composed by pluronic F127. We have a lot of experience with this polymer. It's a biocompatible, biodegradable, it's non-immunogenic, and it's safe in vivo. It's already approved by FDA for human use. And moreover, this polymer, it's easily modulable and allow a low cost and easy production method of the micelles that can be perfectly scalable for industrial purposes. With this type of micelles, we have a huge versatility since we can encapsulate hydrophobic drugs or hydrophilic drugs, or in that case, antibodies that will be treated as an hydrophilic cargo. Also, by adding a cationic polymer, we can encapsulate genetic material. By the simple modification of the hydroxyl terminal groups of the polymer, we can functionalize the micelles with any kind of targeting moiety. And finally, a different application that it's out of the scope of this presentation is that with this polymer, we can also produce thermoresponsive hydrogels for the sustained release in situ of different cargoes and for the cancer local treatment. Using these polymeric micelles, we already achieved the intracellular delivery of antibodies for different targets: the structural maintenance of chromosomes protein 2, SAMC2, that we efficiently transfect into breast and colon cancer cells, and the well-known KRAS oncogene that were able to be transfected into colon and pancreatic cells, as you will see. Regarding the SAMC2 target, I will not stand in detail because it's a work that it's already published, so I will explain it only very briefly. So basically, we conjugated the SAMC2 antibody to the polymeric micelles in two different forms. One encapsulated into the hydrophilic part of the micelles, and the other one covalently conjugated onto the micelles surface. And we use the same micelles to encapsulate conventional chemotherapeutics, such as paclitaxel and cinchofluorouracil, in the inner core of the polymeric micelles to do some combinatory studies. And this is why the objective of the present work and the majority of the research works we have in our group consists in the targeting of both bulk and cancer stem cells. Due to the interconversion model described for cancer stem cells, the therapy would only be complete if we can eliminate both types of cells. Since MC2, it's a component from the condensing complex that it's essential for the stem cell survive, we expect that in cancers, MC2 overexpression, it's associated with cancer stem cell survival and malignancies. So in this work, we combine the MC2 antibody to target more the cancer stem cells, and we use the conventional drugs to kill the bulk tumor cells. This is the physical-chemical characterization of the formulations. As you can see, we obtain a mean diameter of around 35 nanometers with a low polydispersity index, as you can confirm also in the electronic microscopy images. In general, the surface charge of these types of micelles is slightly negative or neutral, and of course, we also confirmed the conjugation of the antibody by infrared spectroscopy. And here I'm showing a representative in vitro result of this project. In this image, we can see clearly that when we use the MC2 antibody conjugated into the polymeric micelles, we obtain a strong inhibition of the cell's colony formation ability. That is a characteristic from the cancer stem cells, as you can see in the graphs and also in the photos. In this paper, we did another kind of experiments to see the efficacy of the particles. Also, we performed combinations with conventional drugs that were encapsulated into nanoparticles core, as I told you. But since I do not have time to show everything, I prefer to show the results that we are obtaining with the KRAS inhibition. In our group, we are very interested in KRAS because it's one of the most prevalent oncogenes in human cancers. The KRAS mutations are present in more or less 90% of the pancreatic cancers and 30 to 40% of colon cancers. And these are the cell lines that we are going to use. And the KRAS mutations cause a constant hyperactivation of the KRAS cascade, which promote an abnormal cell growth and consequently the tumor development. Until today, there are no therapeutic strategies that are completely efficient against the KRAS mutated cancers, and that is why it's still considered very challenging target. So what we did in the lab, since we saw that we were able to deliver intracellular the MC2 antibodies, we tried to do the same with the KRAS antibody. At this point, we are using a general commercial antibody. The idea was if we see some kind of tendency, we think about design specific antibodies or nanobodies for each type of mutation. So we encapsulated the antibodies in our micelles, as explained before. The physical chemical features of these micelles are very similar to the ones obtained with the MC2, so I'm not showing here. And here I'm just presenting the internalization results. In this experiment, we labeled our nanoparticles with a fluorochrome, and we saw that they can perfectly enter in different types of colon and pancreatic cells. And also we saw that these nanoparticles, due to their pegylated surface, they can escape from the macrophage's phagocytosis, which is an important data for the in vivo administration of these formulations. These results are in internalization percentage, and the nanoparticles only enter 30 to 40% in the macrophages, while in the other cells it's around 90% at 4 hours, for example. In this other internalization assay, instead of labeling the polymeric micelles, we encapsulated a fluorescent antibody. And we can see that the free antibody cannot enter in the cells, but on the other end, when the conjugated antibody, yes, it's internalized in the cells. Are these green dots that are inside the cell, the membrane, it's labeled in white, and the green dots correspond to the antibody. So we treated our colon and pancreatic cells with these polymeric micelles encapsulating the KRAS antibody, and we assessed the cell viability after 72 hours. We tested the effect of the micelles in KRAS mutated and KRAS non-mutated or KRAS independent cell line. As you can see in the graph, the free polymeric micelles do not cause any type of toxicity. Regarding the polymeric micelles with the KRAS, we saw a drastic cell proliferation inhibition in the HCT116 KRAS mutated cell line that you can see here, and in the case of the HCT8, that it's the non-mutated cell line, there were no effect observed. So we have a kind of specificity for the mutated cell lines. The same pattern was observed for the pancreatic cancer cells. In the case of KRAS mutated cell line MIA-PaCa2, it was observed a higher anti-proliferative effect than in the PANC1 cells that are KRAS independent cell lines. However, in this cell line, the difference it's not so drastic as in the colon cancer cell line. So we will do more studies with the HCT116 cell line, as you will see. These assays were performed in attachment conditions. For the KRAS mutated cell lines, we repeated the assay in low attachment conditions to see the ability of PM-KRAS to inhibit the colony formation capacity. And in fact, we have a really strong inhibition of the HCT116 colon cancer cells' ability to form the colonies, as you can see here in the graph and very clearly here in the image. And we saw the same for the MIA-PaCa. You can see here a really decrease of the colony formation, the formation of colonies, and here in the photo too. In the cell line, we obtain a little bit of toxicity of the empty formulation because the colonies of this type of cells are really sensitive. And these days, we are doing tests to see the affectation of the KRAS biological pathway after the treatment with the micelles. And from the results that we have until today, we already can conclude that the KRAS and several of the most important players in the KRAS cascade, such as the PI3 kinase, the KT2, and the mTOR, are being downregulated after the treatment with the formulation. The downregulation of BRAF, despite it's not seen at the gene expression level, it is seen at the protein level. So, given the promising results that we obtained in vitro, this made us to decide to do an in vivo proof of concept. So we treated the mice that were previously subcutaneously injected with HCT116 cells, the cell line in which we saw the clearest results, with the PM encapsulating KRAS and empty PM. And we did this 3 times a week during 3 weeks. And as you can see in the tumor volume curve, there is a difference in regarding the tumor growth between the treated and non-treated groups. And also, when we collect the tumor samples, we saw that we have a decrease in the phosphoric that it's also an important player in the KRAS pathway. So, as a conclusion, what we have until now, it's a formulation with adequate physicochemical properties that is able to complex and deliver intracellularly antibodies. Moreover, we obtained a clear inhibition of cell proliferation and colony formation in vitro and an inhibition of the tumor growth in vivo. So further experiments are being done in this sense. We are also planning to do combinatory studies using the KRAS antibody and conventional drugs in the same micelles, and also use more specific antibodies for each kind of mutation. So I hope to have the opportunity to explain you the advances of this project in the future. I want to thank a lot to all the elements of my DDT research group. Without them and our OS institutions and our founders, nothing of this were possible. And of course, I want to thank you for your attention. If you have any doubt, we can discuss or write me and follow us on the social media. Thank you very much.
Thank you very much, Diana. I'm checking and I have not seen any question yet. So then I will suggest that we move to the next speaker. And for sure, please stay with us because I'm sure that there will be something to be asked to you. So we move to the next speaker is Gert Storm, also from Utrecht. And he is going to discuss the spleen targeting of liposomes for cancer immunotherapy. Please make sure that you are in presentation mode, because despite that I was saying to Raymond that it was good, I think there was, okay, this looks good now. Please, go ahead.
I need to unmute. I did. I think you hear me now?
Yes, we do.
Okay, thank you. And I think my topic closely relates to what has just been presented by Raymond Schipperles, although these are two complete separate projects. And it's also about spleen targeting of liposomal antigens in cancer immunotherapy. And let's see if we can move on to the next. Yes. And actually this project fits very well with a sort of recent research direction change my team made in the recent years. And this is the major theme for now for many projects. It's actually for nanoparticles and build therapeutics on nanoparticles for drug targeting to well-accessible body sites. And of course, the keyword here is maybe well-accessible, and as opposed to poorly accessible. Like many of us, and also myself, we focused a lot over the years on getting into tumors, and into the brain is very popular now, but also sites of infection, sites of inflammations. And if you are doing very well, and a few percentage of injected dose, maybe up to 5, 6, maybe maximum 10, but that is very high, is ending up there, and then actually, at least by virtue of the so-called EPR effect. In terms of well-accessible and well-accessibility after administration of nanoparticles, you could wonder, why are we not going to give the nanoparticles directly into or near the disease site? And of course, we know all this is only possible in limited cases, but we started a project. It's not actually dealing with the topic, but just to give you an idea for the eyes. So ocular inflammation, this is a uveitis patient. You can see the inflammation clearly. And based on the results of a PhD student from Singapore, as an ophthalmologist, he did a PhD in Utrecht University. We have also, that's a disclosure to make, recently founded a company, Eden Ophthalmic, in Singapore. And it is this PhD student, Dr. Chi-Wei Wong, who did the work, and this is the co-supervisor, Professor Tina Wong. Anyway, the inflammation, ocular inflammation, is widespread in ophthalmology. And not only in cases like uveitis, but also after surgery, ocular surgery, for instance, cataract, glaucoma, whatever. We focus particularly on cataract surgery. So after surgery, then the patient goes home mostly with a lot of inflammation because of the damage introduced by the surgery. And then, the patient has to instill and apply eye drops containing corticosteroids. It's the most popular treatment. Has to do that maybe 3-4 times a day for a month. And of course, there are drawbacks with such eye drops, limited bioavailability, less than 5%. But also it's often difficult to instill, patient compliance, elderly and maybe young kids have troubles with this. So what we did is different. And this was studied in this paper as part of the thesis. We used liposomal steroids for the treatment of ocular inflammation. And this was in a uveitis model. And these liposomes were given, that's important, subconjunctivally. And now here you see what's being done then. Here is the cataract eye. Then the lens is taken out and a new lens is brought in. And you see there's a complete other reflex here. This is with the new lens. And then at the end of surgery, and we did a clinical study already, then we injected the liposomes containing steroids subconjunctivally. And that is actually outside the eye. So outside, but near the eye, but it's outside. And what the result was in this clinical study, that the patients do not have to use the eye drops anymore. Totally not. You stop the ocular inflammation directly at the beginning, at the time of surgery, with a single injections of liposomal corticosteroids subconjunctivally. So this company is now focused on the further development and clinical work on this, and actually also other types. There are many ophthalmology indications to use liposomes, not only containing steroids, to bring them to the patient. Well, that is direct administration. The other thing, of course, mostly we are stuck with systemic administration. And this is an old slide from Alberto Garbison already, and from the end of the 80s. We know that the liposomes in this case were, but nanoparticles in general, will end up massively in the liver and the spleen, also just elaborated on by Raymond Schiffelers. And over the years, myself too, but many of us are working on avoiding the liver and the spleen to get into tumors and so on, stealth systems. And so relatively little has been done. Now with Onpatro, there's maybe a different situation. Maybe amphotericin B is a different, is an example, but relatively little has been done to exploit this natural tendency of nanoparticles to go to the liver and the spleen. So we work, and this is in Singapore, on fatty liver disease. And many of you may see this picture daily, I don't know, but and many of us might also have a fatty liver and form a certain stage of fatty liver. Caused, that's the general thinking, by eating so-called bad food. And this is the team I'm collaborating with. It's at the NUS, actually in the hospital, the Department of Surgery. And this is Dr. Jong-Wei Wong. He is leading the team, and I'm happy to collaborate intensively with this group in Singapore. Also, Raymond Schiffelers and Martijn Evers are involved, and particularly for constructing the LNP particles. And now, this disease is quite common in Singapore. About 40% of the Singaporeans have a certain stage of fatty liver disease. But it is not only in Singapore. You can see that in Europe, there is also, where is Europe? It's here around 25% prevalence. So it's really a metabolic disorder, which is quite common. And the idea is that, via bad eating habits, we develop a fatty liver, so build up a fat in the liver. At certain stage, there is so much lipid in the hepatocytes, they ballooning. They get damaged, the immune system recognizes that, and then you get an inflamed liver. This stage is called NASH, non-alcoholic steatosis hepatitis. And then if this advances the inflammation, also the fibroblast might get involved, and you get fibrosis formation. We all believe that these stages are reversible. So any drug treatment might help in this. But if you end up with too much fibrosis and so on, you have a cirrhotic liver, then you have liver failure, or you have hepatic liver cancer, then it's too late. Either you need a transplantation or something like this. So this is the context of the fatty liver disease. And there is no approved drug therapy yet, although some drugs are close, are in phase 3, but generally there are quite some side effects associated with the use of these novel development drugs, pipeline drugs. Anyway, what we are doing is to get into the hepatocytes. It's like the petro mechanism via the fenestration in the space of this in the sinusoids and get them into the hepatocytes. And it's not the topic of my talk, so I move on. But with 2 lipid formulations already, we get in a NASH model this type of effects. We can strongly, this is oral staining for fat, we get a strong reduction of the buildup of fat in the liver. It's promising, but it needs a separate talk to go in detail with this. And then the real topic is, but I do it also in a bird's eye view, is the spleen. Disease therapeutics, in this case, immunotherapy, immunotherapeutics to utilize the tendency of liposomes to go to the spleen. And this is with the group in Amsterdam, Dr. Joke Den Haag and Haan and Professor Yvette van Gooik. And these are the PhD students, Martin Nijen Twilhaar, and this is my PhD student at Pharmaceutical at Utrecht University, Lucas Zentner. One major problem in cancer immunotherapy, despite major progress too, is that they often fail due to a lack of sufficient activated T cells in the tumor. And what we try to do is to improve that. So going from a cold tumor, and checkpoint inhibitors fail then because there's a low amount of activated T cells in the tumor, going to a hot tumor, more activated T cells in the tumor, and then the checkpoint inhibitors give a better response. And how we want to accomplish this is with a new liposomal vaccine. And this is the vaccine, is liposomes. You can put the tumor antigen inside or somehow in the liposomes. If you want, you can also put an adjuvant there. But a particular characteristic in this case for targeting certain cells in the spleen is not by changing, like Raymond Schiffelers did, the lipid component so much, but by putting a ganglioside, in this case GM3 gangliosides, in the bilayer. And why are we doing that? Because then we can target to the CD169 positive receptor macrophages in the spleen. And they bind very well to gangliosides, and somehow we can then deliver our antigen to the dendritic cells, particularly to dendritic cell type 1, and this leads to T cell activation in the spleen, and ultimately to hot tumors also in the tumor. This is the concept behind this. And if you look at the spleen, this is the spleen, and don't look at this, is too complex for now, but you see the red and the white pulp. So we are targeting to these marginal zone macrophages, the CD169 positive macrophages of the white pulp at the edge with the red pulp. So we are not so much targeting the red pulp macrophages, which just take up the aging red blood cells and so on. No, and you can see here that with fluorescent staining that we are targeting these cells at the edge between the white and the red pulp. Well, if we do so, then these cells, these macrophages will deliver the antigen to the CDC, the dendritic cell, and a subtype of it is the CDC1 subcell type, which is our major focus, and that will give an efficient activation of the T cells. And what we did first is modulate our liposome system a bit. And you see here that we enhanced the amount of GM3 in the liposomes, and it seems that 3 mol% is optimal for sort of sweet spot for targeting the spleen and targeting the CD169 positive macrophages. You can see here, they are all going here, but the dendritic cells in the spleen, and we also isolated several subtypes of immune cells, are hardly taken up. They are only taken up primarily taken up by the CD169 positive macrophages. Well, we also veritize, and actually for targeting this subcell type, it doesn't matter so much if you take 200 or 400 nanometer. I was expecting 400 nanometer would be better, splenotropic, the larger the size of liposomes, normally more go to the spleen, but for this subcell type targeting, it doesn't matter much. And we are almost at the end of my talk, but we could also see, and we are using an OVA3 ovalbumin-derived long synthetic peptide as the model antigen that the CD8 T cell response is really improved by putting gangliosides in the liposomes and using this targeting to the CD169 positive macrophages. So this is what we want to do with this system. We want to inject intravenously, going to the spleen, going to these edge macrophages, so to say, the marginal zone macrophages, then get T cell activation. And of course, our next steps are very challenging. We have to do in vivo immunization studies now with a real antigen, tumor antigen. The selection of the antigen is also very important, I think. And we have to combine this also with immune checkpoint inhibitors. That's our next step. So this to end up, this seems simple. We just build therapeutics on liver and spleen targeting, but of course we will also face the major obstacles which are usually involved in clinical translation. But I think the NUS, the Department of Surgery, has created a new nano institute and is very much focused on translational medicine, that this could be a big plus here to further develop this. And finally, my last slide, my pharmaceutics team in Utrecht University, my home base for so many years. Thank you.
Thank you very much, Gert. We are very good on time. Nevertheless, there are not questions coming, and I think that everybody probably will come at the very end. So then we continue moving, if you don't mind. The next presenter is Christoph Alexiou from the University Hospital in Erlangen, and he will also talk of immunotherapies for cancer, but this time will be using iron oxide nanoparticles. Christoph, please, I think, Gert, you need to stop sharing.
Yes, let's see how we do that. Yeah, I think I did now.
Yes.
Yeah, thank you.
Okay.
So you see my presentation?
Yes, this is
Thanks
good.
a lot. Okay, so dear Mr. Chairman, thank you very much for the nice introduction. I think this perfectly fits to the slides Gert Storm, the impressive data Gert Storm showed. My talk is about concerning cancer immunotherapy using iron oxide nanoparticles. To go forward, what you can see here is if you do have an infiltration of CD8 plus T cells in cancer, you have a very good prognosis. You can see it here. You have a very good prognosis. If you don't have such an infiltration, often you have a bad prognosis. And so therefore, our aim should be how to really get the T cells, as Gert mentioned also before, into the tumor region. What we are doing in Erlangen is the following. We are dealing very much with iron oxide nanoparticles. What we did in the past, the drug targeting, imaging, etc. What we do now also is that we try to place the nanoparticles into T cells, or the T cells to incorporate the nanoparticles, iron oxide nanoparticles, and they can be concentrated via an external magnetic field, as you can see here in this tube. This is just for a paradigm how we act with the particles. Now, since this is in vitro, we have a clinical unit. A clinical unit. You can see here we do have now the worldwide biggest magnet placed in the robot. I showed it previously. And what we can do is to really concentrate these particles in this tube. As you can see, this is in the clinical operation room in our unit. And we want to really do this, hopefully in the future, also in human beings. But this is a long way due to the fact that we have overcome the following. What we do in our research group is we do synthesis of the particles. We are addressing nanotoxicology, biological mechanism. We do have, as I showed before, this operation theater where we can really do animal experiments, hopefully in the future also human beings and cancer patients. What we are now developing is a pharmaceutical GMP production unit together with partners in Saarbrücken. They do build up now a new pharmaceutical department where they can share a room or 2 rooms to produce the particles in a GMP manner to really do them clinical trials. Now, what you can see here is what we did is that normally T cells don't incorporate anything. But due to electroporation or specific placement, you can see here that the T cells can incorporate nanoparticles here. This is the T cell, this is the nucleus, and the T cells can incorporate these nanoparticles. This is the bars are here 500 micrometers, and so you see the nanoparticles. And the content is around 1 picogram ion content in one cell. Now, the biggest issue is now how to really investigate whether the incorporation of these nanoparticles to influence the T cell modality. And what you can see here, we have here T cells which were stimulated 72 hours after were stimulated with a CD3, CD28, and CD2 activation mode. And these T cells received then after 72 hours iron oxide nanoparticles. What you can see here, it could also be after 72 hours, they could be stimulated. The particles, the cells with the incorporated nanoparticles. Beside this, you can see here also the T cell proliferation. What we have done here is also, we did stimulate the particle. Here you see the control, the cells with spion, control stimulated and with spion stimulated. What you can see here, that after 120 hours, you can see here that the controlled group here can be stimulated, and the particle in the T cells with the particles have appropriate in the same manner. So there is no difference between the control, the untreated T cells, and the T cells incorporated with iron oxide nanoparticles after stimulation. This is also shown here on the right panel with the intracellular cytokine production with CD4 and CD8. There is no difference between the control and T cells and the T cells which were stimulated incorporated nanoparticles. On the other hand, you can see here polyclonal activation to see whether the expression of activation markers on T cells after polyclonal stimulation will be altered in T cells which have incorporated these nanoparticles. You can see after 72 hours or 120 hours, you can see here, this is the control group, this is the treated group. There's no difference between the control group, the untreated without nanoparticles, and the one who received the particles. This is very important that you can see that there's no difference. On the other side, we really got into deeper investigation to see whether the cytokine production of T cells after antigen-specific stimulation is altered. What we did is an MRI exploration of isolated human T cells with peptide-specific TCR. And what we did then subsequent was a loading of the spions 4 hours afterwards and incubation with the target cells. What you can see here is that this is a stimulated control group, and this is a stimulated group of T cells which received or which incorporated nanoparticles. And you can see here in the CD8 different expression modes. There's no difference between the control group and the particles in the T cells which had already incorporated nanoparticles. Next slide, you see here another investigation tool, the proliferation and expression of activation markers on T cells and after antigen-specific stimulation. You can see here the proliferation rate, here in the control group, and here in the cells incorporated the nanoparticles. In all the three figures shown, there was no difference between the control group and the particles in the T cells which incorporated the particles. This is very special because after 72 hours, the stimulation was done and there was no difference in the stimulation mode. Since immunotherapy is a very complex process and a complex system, we investigated whether the antigen-specific killing of target cells will be altered in T cells or after applying T cells, comparing the control in the T cells which receive the particles. And what you can see here, we have here approximately the same cell death. This is a cell death. This is apoptosis in the green are the living cells between control and the T cells which receive the already the nanoparticles, the nanoparticles. Despite the fact that you can maneuver these T-cell hybrids or immunobots, you can also mention these immunobots, this is very important to see that these cells can be visualized in MRI. And this is very fascinating because they can be visible in MRI, as you know. And what we shown here is we had here Jurkat cells. These Jurkat cells had different concentration of the particles. And what you can see here is if you do, of course, give the cells more particles, you have a distinction of the MRI signal loss. That means you can visualize the cells incorporating the nanoparticles, which is really very important for us as a physician. If you do want to place these cells in the future, hopefully in humans, so you have a visualization control. That means a theranostic tool. You don't have only, you can make a therapy of the one side, and on the other side, you have a diagnostic overlooking via an MRI. So take together, what I hopefully could show you is a loading of freshly isolated primary human T cells with citrate-coated superconducting nanoparticles. They have a size of 50 to 80 nanometers is possible. This loading of the particles can be attracted by magnetic forces, external magnetic forces. We could show this in vitro, and we will show this hopefully in the future was in vivo if we receive the respective funding. And thus these primary cells were not impaired in their immunological response by spire loading. This is really very important, that this spire loading does not alter the immunological response. And on the other side, as shown in the last slide, is that this bio-labeling of these cells are enabling the imaging control by an MRI. Yeah, I went through very fast, so I would like to thank all the sponsors and thank you for your audience.
Thank you very much. As I said, we are very good on time, and now they start to come some questions, so we will have everybody at the end of all the presentations. So we move now to the next speaker, and this is Hoda Alavissadet. I hope that I spell the name at least close, from the University Medical Science in Mashhad. And she's going to talk about also novel liposomes approaches and therapies for colon cancer. Please, the screen is yours.
Thank you. Let me share my screen first. Okay, do you have my presentation?
Yes.
Yes, it's okay. Okay.
Yes, it's perfect. Thank you.
Okay. Thank you very much. Good evening from my side in Iran, and thank you so much for the kind introduction, and thanks for the opportunity. I'm extremely happy to be part of this summit. I'm going to talk about the novel strictly stabilized nanoliposomes based on cisplatin carboxylate conjugate for colon cancer therapy. To go forward, first let me briefly touch on the overview of our work. Cisplatin is a key drug in the chemotherapy of many cancers. However, in many occasions, it is necessary to discontinue the treatment due to the adverse reactions, including nephro and neurotoxicity. So, to solve this problem, one way is develop platinum analogs, including carboplatin and oxaliplatin, that were developed to overcome the disadvantages, and they are now the main treatment of ovarian and colon cancers. However, cisplatin is still the mainstay treatment for many solid tumors. One solution is developing nano-based drug delivery systems which offer better selective accumulation of the drug within the tumor and could reduce the distribution of the drug into the normal tissues. To date, we know that EPR phenomena is the primary concept of nano-based drug delivery to the tumor. It is indeed a very well-known phenomena. Just to refresh our memory, EPR is based on the leaky blood vessels of the tumor, which let the nanoparticles to pass through and accumulate within the tumor area. And Doxil is the best example of a nanoparticulate system that perform based on this phenomena, which was thoroughly discussed today by Professor Barnholz. Currently, we can see the vast application of nanotechnology, which enables a multimodal treatment. And further, this technology nowadays is a critical asset for improving diagnostic techniques and also bioimaging of the tumor. Anyway, EPR is not the end of the story, since there are many physiological obstacles that prevent nanoparticles from reaching the tumor area. And the difference in the cancer types can further complicate this issue. So first generation nanomaterials greatly improved the therapeutics, and the next generation were designed to further target the specific moieties or triggering drug release, as well as novel immunotherapy approaches. Regarding cisplatin, a variety of nanocarriers have been studied, including polymeric micelles and conjugates, dendrimers, liposomes, as well as many hybrid nanoparticles. Among them, some demonstrated promising results, and just few have entered clinical trials. As you can see here in this table, half of the formulations were developed based on liposome technology. And here in this slide, I have summarized the main three formulations of cisplatin that entered the clinical trials, but mostly failed the clinic. On the left side, you can see SPI-077, which was the first liposomal cisplatin that entered clinical trials. And the lipid composition was similar to that of Doxil. Multiple trials confirmed its low toxicity. However, it failed to demonstrate efficacy in complete drug release following localization within the tumor area. The next was lipoplatin. In lipoplatin, the electrostatic interaction between the positive platinum and the negatively charged DPPG containing liposomes was used to promote drug loading. This formulation was tested in a number of phase 2 and 3 clinical trials in combination with other chemotherapeutics, but it didn't improve the therapeutic efficacy compared to cisplatin in multiple cancers. And on the right side, you can see liplasis, which is a more recent phospholipase-sensitive liposomal formulation of cisplatin. It was designed for triggered release mechanism in the tumor. However, it actually caused significant renal toxicities and infusion reactions in phase 1 clinical trial, which led to the cessation of the clinical investigation. So we can see that 6 decades after the first description of the liposomes by Alec Bankham, liposomes have not yet been successful regarding platinum drugs. Previously, I've done several investigations regarding liposomal cisplatin by using various lipids of different transition temperatures, using targeting formulation, or as well as triggered release mechanism like thermosensitive liposomes, that all have shown better therapeutic response compared to cisplatin. In fact, the physicochemical properties of cisplatin, including poor water solubility and also the low lipoproteins, drug loading and release kinetics. Recently, Nanocarrier company developed a micellar delivery system of cisplatin composed of a copolymer of PEG, polyethylene glycol, and polyglutamic acid, which use the chemical complexation of cisplatin and the carboxyl side chains of the copolymer to provide drug loading. Here in this study, we address this question of whether using glutamic acids or similar compounds like aspartic acid or calcium acetate could enhance the cisplatin liposomal loading and therapeutic potential. So for this, we have developed various cisplatin liposomes based on the complex formation between cisplatin and carboxylate bearing compounds. Then we have tested these formulations on the cancer cells, and finally the therapeutic potential was analyzed in animal model. To start with the experiment, first we have complexed the cisplatin with carboxylate bearing moieties. And for this, we have used various molar ratios of cisplatin and the amino acids to completely dissolve the cisplatin content. And then we have performed the FTIR analysis to assess the interaction. The peak shift of FTIR spectra demonstrates the formation of a coordination bond between platinum in cisplatin and the oxygen in carboxylate bearing molecules. And we have observed that the peaks related to the physical mixture of cisplatin and carboxyl bearing compounds changed in their complex counterparts. Next step was preparing liposomes. Here you can see the cartoon of the liposome preparation methods we have used in our studies. We have used two methods. In the first one, to develop liposomes, the lipid film was hydrated with the amino acids solutions, and the equilibration method was used for drug loading. So incubation of the preformed liposomes was done with cisplatin at different time and temperature. But in the second method, the complex of cisplatin and amino acids was first prepared, and the resulting solution was used for hydrating the lipid film. Here, as you can see in this table, the average diameters of the various liposomes ranged from 130 to 200 nanometers with narrow size distribution and PDR range of 0.1 to 0.3. The liposomes also showed negative zeta potential within the range of minus 15 to minus 20 millivolt. Cisplatin encapsulation following dialysis was a little bit higher in the first three formulations prepared based on the equilibration method compared to others. Then we look at the toxicity of the liposomes and assessed it on C26 cells. After 48-hour exposure, all the liposomes, as you can see here, were toxic to the cells, and the toxicity of the cisplatin complexes was lowered compared to cisplatin. The next question was the stability of the liposomes in blood circulation. So the kinetic pattern was studied in the plasma and tissue of biopsy mice following IV administration of the free liposomal cisplatin and also the cisplatin conjugates at 3 milligram per kg of cisplatin. Here in this figure, we can see that free and cisplatin conjugate showed the lowest platinum levels in the plasma within the first hour after dosing, and were quickly cleared. So the platinum concentration reached within the range of 300 to 500 nanogram per milliliter in the plasma at 4 hours after the administration. On the contrary, liposomes showed a higher levels of platinum in the plasma after 4 hours following injection. And the highest platinum concentration at this time was attributed to the first three formulations. In this context, we hypothesized that the stability of the liposomes and their escape from the reticular endothelial system led to the higher blood concentration of the liposomes compared to free drug. Next, we have measured the platinum contents in various tissues 24 hours after IV injection of the formulation at 3 milligram per kg of mice weight. The sample treatment in this experiment was done with nitric acid. A high concentration of drug was detected in the liver, spleen, and lung of animals compared to other tissues at 24 hours following dosing. Further, the accumulation of cisplatin liposomes in the kidney was lower compared to free drug, and the liposomes improved platinum accumulation in the tumor area compared to free drug. We also performed the histological analysis on the three major organs, including kidney, lung, and liver. And the data showed less damage by liposomes over 21 days of the study period, and no significant weight loss was observed in the treated animals. We have also performed the hematological parameters, which are not shown here, just to mention. Cisplatin treatment actually reduced the counts of white blood cells, while these parameters were mostly unchanged in the liposomal treatment groups. For the therapeutic efficacy study, C26 tumor-bearing mice were used. For this, after tumor implantation, liposomes were IV injected weekly for 3 weeks, and tumor size and survival were recorded for 55 days. As you can see here in this figure, the liposomes slowed the rate of tumor growth compared to control animals. The first 3 liposomes prepared based on the equilibration method showed a considerable delay in the tumor growth compared to free drug. And we hypothesized that the first method based on this equilibration could better retain the drug in the blood and release it upon accumulation in the tumor. The survival of animals was also significantly prolonged with liposomes compared to free drug, as shown in this figure on the left side of the slide. The formulation bearing aspartic acid significantly prolonged the survival of animals compared to other groups. Taken together, we have described a novel approach of developing long-circulating liposomes based on cisplatin amino acid conjugates. We have compared actually 2 methods for cisplatin loading based on equilibration with the preformed liposomes or the lipid-firm hydration method. The liposomes drug loading, but actually was based on the formation of platinum conjugates with carboxyl moieties of amino acids. The liposomes exerted more toxic effects on the cancer cells compared to cisplatin conjugates, and the liposomes also enhanced the tumor shrinkage and improved the survival of animals. With this, I would like to close my talk and thank the people who helped. I'm thankful to the head of our group, Professor Jafari, my colleague, Professor Ghaibi, and our PhD student, Ms. Mirhadi. I also would like to acknowledge Mashhad University of Medical Sciences for providing the financial support. And I would like to thank Gert and the CLINAM team for gathering this great summit. I'll be happy to take any questions. Thank you so much for your attention.
Thank you, Hoda. This was a very clear presentation. As announced before, we will move now to the last but not least presenter to round this session. This will be Jai Prakash from the University of Twente. And he will also tell us how to reprogram tumor-associated macrophages. So, Jai, please.
Yes, thank you very much. I'm the last, so I have to keep, the attention of everyone. First of all, I would like to thank Gert and the team to put this together, and I hope this is the last virtual conference, and we can meet back in Basel again. My topic is actually, starting from the first, or I would say the second lecture, already on the training, the immune system, and we have heard about, the tumor-associated macrophages, liposomes, all over the conference. And once we are reaching, I'm just trying to bring, some new flavor here. So we are actually designing the liposomes based on the tail flipping mechanism of the new lipid. And for the last couple of years, we have been doing this research. And I would like to give you the flow. I won't go in the tumor microenvironment everybody has talked about. Just to briefly tell you about tumor-associated macrophages, they play a wonderful role in the tumor microenvironment. They cause immunosuppression, even cause pre-metastatic niche formation, and many more mechanisms. I think, again, it's so much discussed in this conference that I would just, keep this part as least as possible. And I would move, to the little bit more details in the macrophages. We know they are M1, M2, broadly speaking, although there have been, subdivisions of M1 and M2. What's more important is that these TAMs, which are the one used, they are largely the one M2 type. And these are the proto-immunogenic. They express a lot of receptors which have been used in terms of either targeting or for modulating them. And IL-4, IL-13, these are the cytokines which turn them into more M2 or TAM-like. What's important is that, these macrophages are the scavenging machine in our body. They just eat. They are hungry. They keep on eating. They eat the dead cells. They eat, oxidized lipids. That's how they detect. So as you can see that they are all active very fast, engulfing, all the particles and the debris in our body. How do they do that? They actually overexpress, the scavenger receptors, and that detects oxidized molecules. Also, the fat droplets here and HDL we heard for the trained immunity from Mulder's group, and they overexpress. What is important is that it's just not a few scavenger receptors. This is a big class of scavenger receptors. These are 8 scavenger receptor classes, and in each class there are number of receptors. What we did actually in the couple of years back, we looked for the phagocytic receptors in M1 and M2 differentiated macrophages. What we found that there were number of the genes or the receptors which were overexpressed in M2 differentiated macrophages. And here we focus more on these 3, CD26, COLEC12, and SCARB1. To look at how macrophages detect the dead cells, they basically detect the surface phospholipids. And if you look at the process, how these phospholipids in the membrane are oxidized, they go through this cycle. I won't go in these details, but eventually during the oxidation process, they make these sublipids, the PAPC, I call it here, and PGPC. What they have this carboxylic chain. It is an SN2 chain of the fatty acid, of the phospholipid. And this is something which these 2 lipids, which we use actually to mimic the system which could be detected by the macrophages. So we made the liposomes, and in which we introduced PAPC and PGPC. And just keep focus on this, this is a long chain, this is a small chain. That's the only difference here. We made number of liposomes with different ratios, and all were about 100 to 115 nanometer size. negatively charged, very good PDI. We confirmed with HPLC that these lipids were present. So we have the CAD detector that can detect, the phospholipids, and these lipids were indeed present in the right amount, which we added. After confirmation, and a lot more characterization, they were quite stable at 4 degrees, while the PGPC, they grew up in size when you put it at 37, while PAPC, they remained stable. We differentiated THP macrophages, monocytes into M1 and M2, and also bone marrow derived monocytes into M1 and M2. But we found that the normal lipids are not taken very well if you incubate only for a short time, for roughly 2 hours, while PAPC were largely preferentially taken by M2, and PGPC, they were taken by both. Probably this was maybe because of the increase in the size. And as you can see in the ratio, like when you have a higher ratio, there is a higher uptake in M2. When we use a low ratio, 10%, 20%, then they are also higher taken up. What we did, we tried to find out how this uptake mechanism is done, whether it's specific or not. We focused largely on these 3 receptors overexpressed on M2. As you can see, these are overexpressed 3 receptors. We knocked down these 3 receptors using siRNA technique. As you can see, it's about 50 to more than 50%. They were downregulated with siRNA, and then we did the uptake study. With the flow cytometry, we checked that when we knocked down especially SRB1, then the uptake was substantially inhibited. We went further. We used human recombinant SRB1 receptor. We coated on the plate using a PA, so the domain for the binding was on the top, and we put different liposomes here. And what we see that HSPC, the plane, and PGPC did not show increased binding, while when we have increasing percentage of PAPC in these liposomes, there was increased binding. So we had indication that it's binding to SRB1. We wanted to investigate mechanism a bit more. So together with our colleagues from University of Twente, we made this model, computational model simulation, where we made this lipid bilayer. This is showing a small piece of liposome, how this works. And if you look at that, these red are the chains. These are the chain, the carboxylic group over here in SN2 chain. And as you see that over the time, and in very short time, within a couple of nanoseconds even, these tails, they flip up. They go in, they go up, and this is the mechanism which we saw over the time. And if you see here, this is largely to focus here that these carboxylic groups, they turn towards the aqueous phase. And this is something which we simulated in different manner, also for PGPC, but this is a larger chain, and this comes on the surface, goes back a little bit, and then goes out. While if you look at just the normal HSPC lipid, the both chains, they just remain in the lipid layer, which is quite imaginable. And if you look at bit more in the molecular dynamic simulation, this is the picture what you see here, and here are these lipids. And these chains, the SN2 carboxylic chains, they tend to be towards aqueous phase. While for HSPC, this the black one, this is SN2, that remains inside the lipid layer. All right, so the next actually is that whether it interacts with the receptor. So we did some molecular docking studies. So we constructed this receptor based on the on other receptor, and we can see that the PAPC is interacting at a very specific place, and this carboxylic chain is binding to the positively charged domain, and this is the arginine which is present, which is positively charged amino acid in this receptor, and it can get into the core and then interact. And there are studies where it has been shown that this receptor and the similar receptors, they can make the dimer, and the liposome actually can interact with this dimer. Further on, that was all mechanistic studies. Then we looked for the in vivo distribution. We had ICG dye or the DII dye in these liposomes. We looked for the biodistribution. What we found, it was all a lot going to the liver, but the PAPC were lesser going to the liver compared to the normal liposomes. The same was valid for the spleen. They were much lesser compared to the normal liposomes. While if you look at the tumor, it was more if you take this ratio, and this was at 2 hours. Here at 6 hours, they were again eliminated from the body, so the signal is lower, but the trend remains the same. It's not only always about getting more to the tumor. It's always important to actually see when you do the targeting whether they get into the right cell type. We did some immunohistochemical or immunofluorescence studies. There we see that there are co-localized in the CD206 positive cells, while in liver and spleen there was less accumulation, which we did also by taking the tissues out, homogenizing them, and looking for the total fluorescence. We also did the flow cytometry analysis, and we found that PAPC was indeed co-localized with the M2 markers. And not after having the biodistribution, now it's a time actually to go for the targeting of the therapeutics. So a couple of years back, we actually investigated, and for the first time, we investigated this STAT6 pathway. And STAT6 pathway is the common pathway for IL-4 and IL-13. These are the cytokines, interleukins, which turn the monocytes into M2 macrophages. And we investigated the mechanism. We have published this, pure biology for STAT6 pathway. So we incorporated this molecule. It's challenging to have hydrophobic molecule into the liposomes. So what we did, we first put this AS compound in the beta-cyclodextrin, and then we encapsulated in the liposomes. We got about 29 or 46% encapsulation efficiency. When we we tested in 41 tumor model, we unfortunately did not find, there was a slight decrease in the tumor growth, but it's just almost nothing. So we were like, disheartened, okay, this is not working. What we looked at, and one of the main mechanisms for tumor-associated macrophages is on the metastasis. So we took the lungs out because the tumor cells, they secrete exosomes, they go to the lungs, and the lungs, they form, there is a reformation, remodeling of the tissues. And when we call pre-metastatic niche, the tumor cells, they already change, the host. They make this tropism for the tumor cells to get in, and that we can capture with the PCR. We looked for, a lot of genes related to the pre-metastatic niche, and what we found that like M2 that was inhibited, M2 marker here, and M1 was a little bit higher, and periostin, SDF, which are the very important marker for pre-metastatic niche, they were completely down back to the normal. The dotted line is showing the normal level of these tissues, and they were really down with this therapy, not with the control or with the free. And the good part is also that the PAPC liposomes did not show any effect on these macrophages. Next, we tried the drug zoledronic acid. We encapsulated it in liposomes and has been used previously. And we see that we can actually kill these cells, M2 specifically with the PAPC, while HSPCs do not show direct effect. If we only incubate for a very short time and examine, the cell viability after 48 hours. We injected in 41 this orthotopic tumor model. We found about 40% reduction, as you can see here, and this was good, but still not sufficient for us. We also looked for the metastatic niche. I'm not showing the data. What we jumped on is the on very important topic is the training the macrophages. We heard about the train immunity for the splenic cells and for bone marrow cells, but one thing is about these peptidoglycans. These can actually train also the macrophages, and these are the small part of the bacterial wall. And if you you use them, they can get into the macrophages, act to the agonize NOD2 receptor, and can train them to be phagocytic and also to kill the tumor cells. And what we did, we used actually miramyl tripeptide, which is conjugated and available with the PE. We put it in the liposomes. And before that, we tested what does it do. And when you put it just on M2, the M2 turns to be more M1. They secrete more TNF-alpha. So these are the M2 cells, and this just you put MTP-PE, and they get activated. We put it in our PAPC liposomes, and what we see that M2 were turning into more M1-like. They secrete IL-1 beta, TNF-alpha. We tested in vivo, and what we found that, with this, we got 70% tumor reduction. We also used another competitor DOPS, the phosphocyrene-based liposomes, that did not show much effect, while our liposomes, they really inhibited the tumor growth. Not only that, we again looked for the pre-metastatic niche, and we also found a very good reduction in all the markers for the pre-metastatic niche. So with this, I think we have come to the conclusion that we have now these tail-flipping liposomes, which can interact with the SRB1 receptor. We showed with the molecular docking, molecular simulations, as well as the direct binding to the SRB1 receptors. We also showed the distribution to the M2 type macrophages, and also we showed that we can train them. Currently, there is a PhD student working on the combination with checkpoint inhibitors. So the first studies, what we have done, that shows promising results. We got synergistic effect, but a lot more to be done over here. I would like to really challenge, because none of this work was done by me. Praneeth Konanthi was a postdoc who did a lot of work. Karan Posma, who initiated as a PhD student on this work a couple of years back. Kunal is now continuing this work. Emma did more computational work, and this is my team. And of course, I would like to thank Peter van Hogevest and Gert Storm being interactive and keep discussing on this project. And I would like to thank all the audience if they are listening to me at this moment. Thank you very much.
Thank you very much, Jai. I recognize how difficult it is to give these talks in 2D, as I always call, because we never have the feedback of the audience. But I hope that there is a lot of participants listening to all of you, and that all the participants, please, they should go now to join the discussion, going to the button that is, I think is the debate room 3. And for all the presenters, please do the same, but do it accessing the link that you will find in the chat. And I hope to see all of you there in a few seconds, and then we will go through all the questions. Thank you.